Welcome everyone again to this second week of the Statistical. So our first lecturer is Akshit Goyal, who is from ICTS, and we'll talk about statistical physics of ecosystem.
Okay, thanks a lot, Abhishek and Sanjib, for inviting me to come. Hi, everyone. My name is Akshit. I just put some things up on the board just to give you a sense of what we'll be talking about today, but I'll just briefly say that over the next week or so, I hope to kind of give you a little bit of a taste and a flavor for what it means to do sort of statistical physics of maybe a very different kind than maybe you're used to sort of studying. And also maybe what it means to be doing like statistical physics in a field that's also very, very new. So statistical physics of ecosystems, which is what I'm going to be talking about to you this week, is a field that's basically about 10 years old, maybe a little bit less, but it's sort of growing over time. But sort of before I sort of get into statistical physics of ecosystems as a whole, I just want to give you a sense that, of course, ecology is not a very new field. That's about maybe, let's say, 100 years old. And that's really, just very, there's, if you kind of study biology or, if you sort of study physics, we sort of look at systems at all scales. Right? So in some sense, biology and biological processes also go at all scales, or all the way down to the level of single molecules and proteins, and all the way up in some sense to what we think of as ecosystems, collections of different individuals that then interact with each other and stay sort of diverse, right? So in some sense, ecology stays at sort of the highest level of organization within sort of biology. And it's really kind of the study of life and its sort of biodiversity, right? If you sort of walk out here or, out in the forest or somewhere, you'll just kind of be amazed by the large number of species of trees and animals and plants that we kind of all see around us, right? And so ecology in some sense poses a rather large question, which is how does it get to be the way that it is? And maybe it sounds a little bit vague right now, but we hope to, my hope will be to kind of make some of these notions a bit more concrete, at least in the context of a few simple models that we as physicists can write down and sort of work from there. Yeah, so that's kind of going to be the very, very broad goal of what we're going to do in the next few classes. As usual, I should say as a disclaimer, please feel free to interrupt me during the class at any point. I'll try to intersperse the class with some sort of active exercises for you to do, some questions for you to answer. So we'll try to keep it as interactive as we can, right? Okay, but then maybe before I sort of talk about even ecology, what I'll say is that sort of ecology itself started from an earlier discipline of sort of population dynamics. And this sort of happened maybe about 150, about 200 years ago, when people were interested for all sorts of reasons in, let's say, livestock populations and so on. And what people started to do was to look at what was happening to the dynamics of different populations, let's say sheep. So people were, of course, very interested in herding sheep and sort of growing sheep for obvious reasons, other animals, as you can imagine. And what people started doing was to not look at individual sheep, but to sort of look at their entire herd or, an entire population of sheep. And so what people started to do was to try and essentially do what we have in India, which is like a census, right? So you can kind of do a census of different animals. Let's say you do a census of sheep. You kind of just try and plot. You just go around and collect the number of sheep that you see sort of in a plot. You do that kind of every year or every couple of years, and you try to kind of plot that, right? And so when people plotted that, this is kind of what they saw. So, we'll have time on the x-axis, maybe that goes in something like years. We'll just plot the number of, again, I'm using sheep as an example here on the y-axis. And what I'll do is, and this is something that's going to keep on coming up again and again in this course, which is that we're going to imagine the y-axis of these plots, the population sizes, go on a log scale. Again, I'm going to ask you in a couple of minutes why that ought to be the right scale that we should look at. But really what we're kind of looking at is just kind of trying to make some, so you go at year 0 or something like that, and you count the number of sheep, you make a point over here, you go over to the next year, plot the number of sheep again. Maybe it's gone up a little bit because you did a little bit better at herding sheep. And you keep doing this again and again and again. First question, why do you think the log scale is a good scale that we should be thinking about on the y-axis? Right, yeah. So in some sense, we're kind of imagining that these populations are growing exponentially, right? And why might that be the case? Why might it be natural to assume that the growth of the population is an exponential process? Right, but even yeah, exactly. So one, maybe two give rise to two, and maybe that's also fine. Maybe two give so right. So if you're imagining that you're reproducing by, let's say, splitting or something like that, one gives rise to two, two gives rise to four, and so on and so forth. That itself gives you an exponential process, right? And so why is it that for an exponential process we'd be interested in making things on log scale? Sorry. I guess any scale, I mean, if the population saturates at some level, it would also saturate on a linear scale, right? It's just we like making lines, right? So if something is growing exponentially with time, what would that look like in a log versus linear scale? It would just look like a line, right? So that's kind of what people saw. That's kind of the first kinds of observations that people had when looking and plotting these sort of populations over time. Maybe this is over the matter of a few years or maybe a decade or so. But do you think this went on forever and ever? No, right? So the second kind of observation that people often saw was that this process kind of stopped at some point. And at some point, the population of sheep or whatever it was reached some value. Maybe there were some fluctuations around this value. And I'm just kind of making these lines over here just to give you a broad sense of what was going on, right? So there's sort of one phase where there was some sort of exponential growth, and then sort of some phase of the population where there was saturation. Right? And this itself was, maybe you might say that it's not kind of surprising at all. That's kind of what you would expect. But in some sense, at the time, people were kind of amazed by this. Mostly because it kind of suggested that if you wanted to look at a population, you may not necessarily have to track what's happening to every single individual at any particular point of time. Of course, things happen. Some sheep die, some sheep get sick, some sheep have more babies than others. But there was some statistical regularity to the patterns of the population size over time. Right? And that's kind of what motivated this idea that maybe we can think about dynamics that happen at the level of populations instead of dynamics that might happen, with us tracking every single individual at all points of time. And this kind of gave birth to this idea of tracking population, of sort of the field of population dynamics. This is not the only thing that people saw that was interesting. There were other interesting patterns that people started to see when they started to observe these things. So another of these patterns was the idea that sometimes in some populations, this was especially true of fisheries. So people were, of course, also very interested in growing fish. You can kind of sense a theme here, what kind of populations people are trying to track over time. So people are trying to grow fish. And what people started to notice was that, again, now if I imagine that my y-axis is the number of fish over time, and at some point I'm going to stop writing log here again and again, over the course of these entire 5 classes. If I'm plotting population sizes, kind of assume that I'm plotting them on a log scale, right? So yeah, what else do you think that's interesting that could happen? Maybe I know for other populations that isn't just simple exponential growth and saturation over time. Oh yeah, okay. Maybe you see oscillations. In fact, yeah. I mean, right now we're just talking about tracking one species over time, but maybe yeah, you could track maybe 2, and you could start to see oscillations. But that's not what happened with fish. And by the way, these fluctuations that I'm talking about here, they're not really oscillations per se. So do you think that if you start with any number of animals here at the beginning, you'll always get exponential growth to start with? Some universal scale or what? You're trying to explain why, like you're trying to explain the slope of this line, right? Yeah, but the slope of this line was not I mean, that's more a quantitative phenomenon. What people saw that was interesting was a qualitative phenomenon. So what they saw was if you start with a very small number of fish, the population actually crashes. Okay? And again, I'm going to stop making points, but really it's hard to draw zero on a log scale, but assume that there's a special point over here that's zero, and really the population crashes. But this is not always the case. If you start with a large enough number of fish, then things will grow and kind of saturate. Right? So there was this idea of a kind of a minimum population size or a critical population size that you might imagine fish might need for them to start surviving. Can you imagine some reasons for why this might be the case? Inbreeding. Can you say more? Okay, so it could be that maybe there was not enough genetic diversity in the population, so something bad happened and they crashed. Maybe this was not the case with these fish. Maybe there's a competition between your death rate and the birth rate. Okay, fine. And so why would that result in a minimum population size? Diseases. Okay, and what's the argument? I mean, you need to say something more than just this word. Yeah. Okay. Maybe there's a predator population. Right. Okay. And why might there be like a minimum fish population that might be needed to be good against a predator? Yeah, maybe you can sort of herd in school, and then maybe once you have a large enough fish population, maybe you can avoid predators better. Maybe there's some sense in which, yeah, there's something cooperative about the population of fish that allows them to do better in slightly larger population sizes, right? Of course, the population size gets larger and larger. At some point, they're all competing for food, so maybe they kind of stop growing. But that might be one reason why this might happen, right? So remember, people didn't particularly know that this was the case. These are kind of stories that we make up, right? But these are observations. Yeah.
Like I've seen in an aquarium that fish they can grow. Like at least
Indeed. So of course, if you like control conditions, so for instance, if it is really the case that predators are one reason why you might have a minimum population size, then if you grow fish in the absence of predators, then you might imagine that this effect goes away, right? And so maybe that's kind of what's going on. So in a lot of fisheries, actually, now we know that there's lots of schooling behaviors that are important. Some of these schooling behaviors, and this is not, every fish is not the same, right? So some fish might have this, some fish might not. Okay, so I mean, are people getting the kind of motivation for looking at these population dynamics, looking at statistical regularities in them, and then trying to see, hey, maybe we can think about models that are sort of broad population scale models. People, of course, also started to think then about two species models. So this kind of effect, by the way, where you have minimum population size, discovered by Alley and is now called the Alley effect. This kind of behavior here, where there's exponential growth with saturation, will often be called logistic growth. People also started looking at two species populations, and this is now about 80 years old or so. These experiments were done by Gausser in the 30s. He was really now looking at really tiny populations, like microbial populations of protists and little things that he could kind of grow in the lab in like little flasks. So he could kind of really count them over time. Right? All of these data, by the way, coming from counting expeditions. Here again, we look at time. And now we're looking at time in number of days. And here again, he's looking at number of individuals of a species. And really what Gausser started to do was he started to mix two species together, right? We can, for our intents and purposes, we can think of them as species A and species B. And so if he grew them together, what he saw was that sometimes when he would mix two species, A and B, he would see that maybe over time they would kind of grow, and at some point they stop growing. But at the end, both species A and species B are both alive. Okay? So in some sense, they're coexisting. Yeah. So he didn't know whether they were prey-predator type or anything. He just literally went to his pond, picked out, scooped out a bunch of things. He isolated a bunch of different protozoa that he could see with his microscope. And then he put two sort of random ones together. Okay? So we don't know whether they're predator or prey. We don't know whether they're competing. We don't know whether they're cooperating. We're just setting up what he saw. Right? We'll talk about how you then explain this stuff, right? But not every pair of species A and B he mixed up had this kind of behavior. Sometimes when he mixed species A and B, what he would see was that maybe one species went up and saturated, and the other one maybe went up initially, then went down and crashed. Maybe this might be some species C, some species. And so in this case, both these species, are they coexisting? No, right? Maybe transiently there's some period at which they're both present. But if you wait for a long time, species C will outcompete and exclude species D. This phenomenon is called exclusion. Again, remember, Gauss didn't really know why this was the case, but this is what he observed. Okay? There are, of course, other kinds of observations about hare and foxes that you might have heard of, predator-prey, where you start seeing oscillations between two species. That one maybe you've heard about so much that I won't really draw it again. But I'm just trying to get you motivated to thinking about population dynamics going up from a single species. So already in some sense we are statistical, right? We're not thinking about individuals. To two species. And this is kind of what went on, at least experimentally, for a long time. But more recently, what has started to happen, and like you can imagine that these experiments are hard, right? The experiments themselves are easy, but collecting this data means going out there every single day, extracting whatever species there are, and literally counting them, right? This is extremely tedious. Now you can imagine trying to do this with trees or with plants, identifying, let's say you go out to a rainforest and you want to do a census of different species in a rainforest. How many species do you think there are in a rainforest typically? Are there 1, 2? Thousands, right? And so first of all, you need someone who can tell the thousands of species apart. And then second of all, you need people who are highly trained, highly skilled, like you and I would not be able to do this, right? To be able to go and look at every single tree and first identify which species it belongs to, count it, and then go to the next tree, identify which species it belongs to, count it, mark all the trees so that you're not double counting, right? And then do that kind of every year or every 5 years and so on and so forth. It sounds ridiculous. This is indeed one of the biggest ways in which we had quantitative data in ecology for the longest time. There are these Smithsonian research centers where there's entire islands near the United States where they would grow trees in like 50-hectare plots and track them and do a census every 5 years. This still sort of goes on in a big way that we get data. But again, you can imagine that as the number of species goes up, if you want to study actual ecosystems that are out there, this kind of gets a little bit out of hand, right? Tracking things becomes difficult. And what else might become difficult? Let's say you had something perfect to be able to track. Maybe you had a satellite that goes up and counts every single tree, assigns it the right species, and does it perfectly. Is that still going to be a reliable way to get lots of quantitative data? Why is that? Why no? Let's say you have a perfect measurement instrument, No double counting. Let's say you have perfect characterization of trees. Just you have a perfect measurement device. We have big computers, right? There will be fluctuations, but if the fluctuations are real, you want to track that, right? Okay, so one argument is that maybe we can do that for one system, but not many. Maybe, let's say we could do it for many. What about the x-axis of these plots? Time is, yeah, you're waiting years, right? And why is it that you're waiting years? Let's say you have a perfect measurement device, you could just do it every few minutes, right? Sorry? Yeah, exactly. The intrinsic time scale of the actual dynamics is not happening over the scale of minutes, right? The intrinsic time scale of these ecological dynamics, fish reproduce at a certain rate, right? So if you track things at faster than that rate, you're not going to see much. Trees grow at a certain rate. So if you track things at faster than that rate, you're not going to be able to see much, right? So there's a natural time scale, there's a natural intrinsic time scale of these dynamics that limits how much you're able to see, right? So now we're able to see kind of two problems of scaling up these approaches if we want to really study the actually diverse systems that we see out in our world, right? What are they? Yeah. Yeah. Very good. Yeah. Very good. That's actually a really good question. So the question is really about when you go out there and you see a single species of something, is that enough to say that that's there? Yes. And actually, this is not that uncommon an observation. Actually, much of this data set, for instance, that I was talking about on the Smithsonian Research Institute, there is a large number of trees actually that have just one individual, right? That doesn't make them any less. Yeah, so you could, if you wanted, just to look at a single species, you could make that plot for single species. You could also make plots of this kind where every trajectory, every kind of line that I'm making belongs to a species. So then you could, if there are 1,000 species, there would be 1,000 such lines that I would have to draw, right? Some of them would be at some very low number, like one individual. Some of them would be at very, very large number, right? Okay, but to go back to what I was saying, if we really wanted to scale this up and get a lot of quantitative data because we want to understand what other kinds of statistical patterns that might exist when there are a large number of species, we are confounded by time. Trees just grow too slow. Animals just grow too slow compared to the time scale of graduate students and scientists' lifespans. And the second is we don't have this perfect measurement instrument, right? We don't have something that currently can identify species down to the level of individual to individual, right? Both these problems were somehow circumvented or have somehow been circumvented over the last 10 or 15 years. Not in trees, but in something else. Someone guess? Yeah, exactly, in bacteria or microbes. Why could, like, what is it about microbes that could make you solve these two big problems? They grow fast, yeah. And do people have a sense of how fast microbes typically grow? Trees, yeah. Do people have a sense of how fast roughly trees grow? Is it on the time scale of months, year, decades roughly, right? Yeah. And what about bacteria? Typically tens of minutes to maybe an hour, right? So that's already, we're going from decades to hours, right? So we have this big time scale advantage. Though it seems like on the other front, we made it a bit harder, right? Like we couldn't count trees and identify their species. How are we going to be able to look at these tiny bacteria that we can't even see and identify which species they belong? So this problem has also been solved. Someone know how? Exactly. So what you can do for bacteria that you can't really do for trees as easily is that you can essentially crush up samples of like big soups of bacteria. You know, you can take some from your gut, you can take some from the soil. Literally anywhere you touch out here on Earth, you scoop up something, you will find a community of diverse bacteria. And again, just like rainforests, these are not single bacteria that you see, much like what the Dental Association of India would like to try and tell you. These are communities of thousands of species of bacteria. So much the same problem. Thousands of species of bacteria coexisting with each other. It's much faster time scales and somehow an uncanny ability to get quantitative data. So let me tell you just very briefly in the next 5 minutes how that comes about to be the case and how there's been a little bit of a quantitative revolution over the last 10 or 15 years, which is really what's caused the resurgence of this statistical physics of ecosystems, which is what we're going to study. So, I mean, before sort of this, theoretical ecology as a whole has been a very rich quantitative discipline, about 50 or so years old. But essentially what theoretical ecologists have been doing is trying to understand what happens in situations of either this kind, when there's a single species, or situations of this kind, where there are two species. Okay? Maybe sometimes people would go to situations where there are three species. That already kind of gets a little bit out of hand. And the argument was, of course, well, that's the kind of thing that we can get reliable data for, so that's what we should be working with. Okay? These are things that have changed. Okay? So the broad idea of sort of this quantitative revolution in microbial ecology, so to speak, is that you're really able to go out there to any particular sample. Let's say I go out to a nice beach or some nice marine colony. I just scoop out some part of the water because I'm interested in the marine microbial community that forms around it. Like I said, there's microbial communities or bacterial communities literally everywhere around us. What I can do is I can just kind of scoop it out, bring it to the lab. And if I were able to put a microscope to it and kind of see, what I'll see is just an incredible diversity of different kinds of species. I'm really not very good at sort of drawing these things. You'll see many, many different kinds of species. And again, we'll talk about the numbers, but really, like if you look at sort of any of these columns, what you'll see is like roughly 10 to the something cells per ml. Someone hazard a guess what power of 10 roughly we're talking about? Just the densities of how large the populations are. I guess for sheep, yeah, we didn't talk much about numbers, but for sheep, I guess typically it would be in the hundreds, right? That's kind of what someone's livestock herd would look like. What kind of numbers are we talking about for these kinds of microbial communities? 10 to the power what? 10 to the power 10, okay. Any other guesses? 10, okay, so someone thinks a million cells per ml, by the way, yeah? So this is, everyone knows how big an ml is. Okay, so 10 to the 6, 10 to the 10, any other guesses? No one thinks it's 10? No one thinks it's 10 to the 50? 20? Okay, I mean, it's somewhere close to 10 to the 10. Yeah, so that's not, turns out to be for many, many cases, not that far from the truth. And again, like we said, there are roughly 10 to the 3 to 10 to the 4 different species that you see. And how do you tell that there are these different species? Well, again, you can't count all of them. What you kind of do, and I'll gloss over this because I really don't want to get into the details, but I encourage you to look at it if you're interested, is that you have some way of essentially crushing this entire soup of cells and extracting the DNA from within each of these cells. So everyone knows that cells are made of DNA. I mean, they all contain DNA. And so in some sense, what you're trying to do is to extract that DNA, and you want to crush up cells. Does someone know a fancy way of crushing up cells? Really fancy instrument. Has anyone been to the kitchen? I mean, okay, how do you make ginger garlic paste in the kitchen? Exactly. And what are you doing with a mortar and pestle? You're crushing up cells, right? So really all you're doing is you're just using a little mortar and pestle to literally like crush up these cells. And once you sort of crush them up, all their DNA is exposed out. And essentially for all intents and purposes, you throw them into a DNA sequencing machine. The details of this machine I will not talk about. And out basically comes data that you can analyze on a computer. Okay, there's a lot that goes into each of these steps, so we won't sort of talk about it. At the end, you get data for which species are there and at what relative abundances. So you'll be able to get to know that there are, 10% of species A, 3% of species B, 20% of species C, and so on and so forth. Okay, you can do this for this sample. You can do this maybe someone was talking about statistics, so you can go and do this for many, many realizations of these systems. You can go to another beach, or you can take another sample and kind of add this. You can do many, many samples of this kind. You don't just need to do lakes or aquatic systems. You can go to the soil. Everyone knows that soil has plants. You can kind of go deep into the soil. And does anyone know how plants get proteins? Yeah, exactly. I mean, proteins, essentially, they need nitrogen and they can't do that on their own. There are all these bacterial communities that are in the roots of these plants. They somehow assemble and get to these plants, and there's some symbiotic relationship between them. If you wanted to understand what communities they were, you can just go and scoop near the root. You can, if you wanted to know what bacterial communities were inside yourself, you could do that as well. Do we all know we have bacterial communities inside ourselves? Where are they? Nope, just literally everywhere. Right? So they're not just in your intestine, they're not just in your gut, they're in your mouth, they're on your skin, they're literally everywhere. Yeah? Often people will tell you about your gut microbiome, and why might they tell you about your gut microbiome? It aids in digestion. What kind of digestion? Are there some things that we eat that we actually cannot digest? Plants, basically, right? Like we, as humans, we don't possess the enzymes or the machinery to actually be able to digest plants. Okay, so the only real reason why we are able to actually get any nutrition from plants is not because of enzymes that we have, but it's because of enzymes that this huge community of gut bacteria inside us have. And that's why whenever you have diarrhea or something, which is when you kind of void that community, you have kind of trouble digesting. Yeah? Sorry for the gory detail this early in the morning, but you could also then go and sample your own gut bacteria if you wanted, not by going inside your gut, but by sampling what comes outside, right? Okay, so there's some way, which I won't draw here, which is early in the morning, where you can also sample it from human guts. And you could also do it for these experiments of Gauss's. So you could have bacterial communities growing in flasks inside some lab where you can control experimental conditions. You can also go and sample them. And so the same kind of approach and pipeline applies for all these different systems that are out there. And this thing is fast. This thing is fast, it's quantitative. And so that means that you cannot just look at the same sample at time t. You can also look at it tomorrow and day after tomorrow and the day after that and so on and so forth. And it'll actually make a difference because the intrinsic time scales of the dynamics of these communities happen over the course of days as opposed to decades. Right? So that's really the proliferation of these kinds of data sets has really what has made ecology into a much more quantitative discipline than maybe it used to be about 50 or 60 years ago. Okay, and that this really sequencing revolution has kind of caused that. Okay, so really by getting data of this kind, this huge amount of data, what we've been able to do is that we can really study the statistics. of ecological communities. And I think for the rest of these classes, I might use the word communities interchangeably with ecosystems. I kind of mean the same thing, right? It's just some diverse collection of species is a community or an ecological community or a microbial community or a bacterial community. All these things will be kind of interchangeable. Okay, and so by studying these, of course, we need to say something about what kinds of statistics or what kinds of properties we're interested in. And really there are many of them, but there are broadly three that have historically been very interesting and relevant. And these are the three that we're going to focus on in sort of the rest of these lectures. And so those three are the first, which we already started to talk about a little bit, which is diversity. Is how many species are there? How does that number change over time? How do all of these species coexist? Right? The diversity question is really a question about if there are 1,000 species that are in this little sample, and if there are 1,000 also tomorrow and 1,000 day after and so on and so forth over and over time, somehow these 1,000 species are able to coexist with each other for long, long times. How does that get to be the case? And so again, if you sort of do all of these surveys for lots of these different communities, you can now start to make essentially histograms of how many species you would see in any particular kind of community that you go out there. Let's say you sample this marine community again and again and again and again and again, different realizations of it. You can then, make a histogram of how many species you see in these different communities. And with these histograms, it might tell you thing that we were talking about previously, which is that's kind of where you see the roughly 1,000 to 10,000 species per sample, right? So these kinds of distributions are one set of things that we're interested in understanding. That's diversity. The second one is stability. And this is really a catch-all term for the kinds of dynamics that take place. And so what I mean by that is that sometimes when you look at a particular community over time, right? Remember, we're looking at dynamics. You might see, let's say I start making plots of the kind that I was making sort of earlier in this class. But now I'm not going to make them for 1 or 2 species. I'm going to make them for thousands of species, right? Using these kinds of data sets. So here on the y-axis, I'm going to plot this quantity that I'm going to call the abundance of species A or B or C. And whenever I say abundance, that's again going to be a catch-all term for some notion of population size. It might not always be numbers, because even with, as soon as we go to like bacteria and stuff, we're talking about really densities, right? So we're really talking about population densities as opposed to just single numbers. So think about some abstract quantity of that kind that correlates with how many individuals there are. And that quantity in ecology is always conceptually called the abundance. Okay, so abundance is going to be, again, a catch-all term for the population size. Okay? But may not necessarily be an integer number. That's kind of what I'm trying to tell you, right? And it's very easy to imagine how it might not be an integer number. You might just divide the actual number of individuals by some relative scale, right? By some relevant scale of how many individuals you expect. Maybe that would be by how many individuals you typically saturate to. And this is a number. This is usually a quantity that you might have heard of many times. It's called the carrying capacity. Just by kind of dividing by some intrinsic scales of population size, you might get to some continuous quantity of this kind. We're talking about really large numbers anyway. So sometimes what you observe is that really, if I plot the abundance of many species over time, that really is pretty stable, right? I'm not drawing any of the little fluctuations over here. But you really observe the same species, the same abundances over long periods of time. That's really the notion of a stable community. But that's not always the case. sometimes what we observe, this is indeed something we're going to start talking about in lecture 4 or so, is that the abundances of different species are not really this nicely behaved constant numbers over time. They instead exhibit massive fluctuations. So sometimes you might observe that the abundance of a species suddenly shoots up, becomes very large, and suddenly shoots down, becomes almost zero. The species almost goes extinct, then comes back again. Some other species is kind of doing something else, not necessarily in any synchrony with this species. And these kinds of fluctuating dynamics go on forever and ever. So in some sense, you have the same number of species. The diversity is still 1,000 or so. But if you actually look at the dynamics of that diversity, the dynamics will change very, very rapidly, right? And you almost get these supposedly persistent chaotic fluctuations. And this is kind of an unstable community. Right? It's unstable in the sense that there's no fixed point that you're heading towards that then you will stay sort of stably around. Whereas this is kind of that situation. We'll talk about more of these examples when we come to the individual classes. Again, I'm just trying to give you a flavor of the kinds of things that we see. And the final third thing that people are interested in, diversity, stability, and structure. And structure is kind of very simple. Structure is the question of asking, if I go to a particular system, if I go to a particular community, I see many, many different species. Do you think all of the species are going to be at the same abundance, same population size? No, right? Some species are naturally going to be more abundant. Some species are going to be less abundant. And so if you have many, many such species, you could then try to plot and ask, what's the distribution of the abundances that you see in a particular system? And again, you might draw some sort of histogram of that. And again, we see lots of statistical regularities here. And so again, this understanding this distribution of the abundances might also be something that people are interested in. Okay, so saying again, there are going to be 3 statistical properties of communities that we're going to be interested in. One is understanding diversity and coexistence. How do large numbers of species coexist with each other? How many should we expect? Should we expect this number to be 1,000 or 10,000 or something else? We're going to care about the stability of the dynamics of this large number of species. And then, especially in the case where the dynamics are stable, we're going to also care about the structure of the community, which is which species are more abundant, which species are less abundant, and why might that be the case? Maybe you're already thinking about, oh, some species are better, they grow a little bit faster, so maybe they'll have more abundance. Some species are less good and they grow less well, and then they have lower abundances. You will see that if this is your intuition, these ecological models will actually completely break that intuition. That's not the case at all. Okay? There's the broad sort of motivation of what the study of ecology or population dynamics is about, how we get quantitative data, and then what we're interested in here. Excellent question. So people do study spatial variation of populations, and the way that people do it is not necessarily to think about the spatial variation that's going on within a single patch, but instead on a global scale, the dynamics of many, many patches. So we'll talk about that in a second. Okay, so that's all I wanted to say about data for today. So sort of in the next 45 minutes that I have left, I want to do 2 things. One is to give you a broad sense of what modeling looks like in ecology. And the second is to give you a sense today of take one model and try to study it not in this high dimensional, high diversity limit, which is something we'll start doing tomorrow, but instead look at these models in the low diversity, what I call low dimensional limits. So throughout this class, I'm going to talk about 2 things: a low dimensional ecology, which is a situation where there are just a small number of species, 1, 2, or 3. And then a high dimensional ecology, which is an ecology with a large number of species. And in fact, that number of species is going to tend to infinity, at least in our models. Okay, so I call them dimensions because you might imagine any community as being a point in some space where the axes of that space are the abundance of species 1, the abundance of species 2, the abundance of species 3, and so on and so forth. So as the number of species grow, the sort of dimensionality of that space grows, right? So we're going to be thinking about high dimensional spaces, and by that I mean infinite dimensional. We're not going to do that today. Okay? All right. Yeah. Exactly. The first one is you go to any community and you ask how many species are there, and you go to another community and ask how many species are there. So this is over many samples of many communities. This is over one sample of one community, but with many species. Okay, and so like the broad thing that we're going to see as we start to understand each of these three properties is that species are not independent of each other. Species are not behaving and acting and growing independently of each other as what we saw maybe in that sheep example, but species are fundamentally affecting each other. There are interactions of all kinds, and really it's studying the consequences of those interactions that ecology is all about. Okay, and so like our goal is going to be to try and understand and explain all of these statistical properties and patterns using minimal conceptual descriptions. Okay, so of course one technique that you might have is in any particular system you go and try and understand what every single species is doing, how they're interacting with each other, and really try to understand that's one system well. We're not going to be taking that approach. We're going to be taking a different conceptual approach where the understanding will be that these systems can come from any kind of environment, right? But what is going to be common to all of them is maybe some sort of universal set of ecological processes that cause the diversity, stability, and structure. And it's sort of understanding that from a conceptual minimal description that we are going to care about. Okay? Not to say that the first kind is not worth doing. In fact, lots of people do it, but that's not going to be the approach in this class. Okay. Let's see how far we get. Okay, so what are the broad theoretical frameworks that we have? One broad framework that we have is just like there was a question right now, what we're imagining is that ecosystems consist of many, many spatial patches, right? There's patch 1, patch 2, or let me actually call this patch A, patch B, patch C. In each of these patches, there's a community of many, many species, you know, just like what I was drawing sort of over there. Of course, I could care about the spatial variation within this patch. We are not going to do that throughout these classes. Okay, that's something that we can talk about later. But this is not what is typically done. We're thinking of a patch as a sort of well-mixed system. Okay, there might be species that go back and forth between these patches. There might be, that is to say, some migration events. Within each of these patches, species might grow, right? They might grow because there might be resources that are available. They might consume those resources. And as a result, they might be able to grow. And then, of course, this growth might not be deterministic. There might be some noise because why might there be noise? There might be noise in the environment. I guess resource availability might fluctuate. But then also when populations grow, I mean, underlying it, remember we were saying that we are not thinking about the birth and death of every single individual, right? We're somehow thinking of a broader description. But of course, as a result of the fact that there are births and deaths of individuals, there's going to be some stochasticity or some demographic noise that is going to come as a result of that, right? And so one might also think about noise, right? And of course, then you're also thinking about the dynamics of different resources, which I'm going to represent by these triangles and squares that might be supplied from somewhere, right? And sort of move around. Within each of these patches, you might think about how species are interacting with each other. And so what we're going to think about is a situation where there are lots of different species. And this is species, 1, 2, 3. There are different resources that might be present in the environment. And these species might be eating these different resources, right? And as a result of that, growing. And so the simplest kind of model that you might be able to write down for the dynamics of these species and resources, something called the MacArthur consumer resource model. Or generally consumer resource models. I'm going to call this MCRM for the rest of this class because it's tedious to write MacArthur consumer resource model. And what you're doing in sort of MacArthur's consumer resource model is a very, like a simplification of this big thing that I told you about. So we're not going to be thinking for now about migration. We're going to focus on just one patch, okay? And we're not going to think about noise. We're going to assume completely deterministic dynamics, okay? And so really what we need to write down, what we are going to write down is essentially some dynamics of each of these species that we have. So each of these species will have some abundance, which we're going to represent as N. N's are going to be the abundances of different species. So N1 is going to mean abundance of species 1, like how many individuals there are, but we're going to think about continuous numbers. And 2 would be the abundance of species 2, and 3 would be the abundance of species 3, and so on and so forth. We are going to want to write down the dynamics of dni dt. That is, there are 3 species, there are 3 such equations. And then we're also similarly going to want to write down dynamics for each of the resources. So again, there might be some resource 1, resource 2, and resource 3, and so on. And I'm going to use the subscript alpha to always talk about resources, alpha, beta, gamma, and so on and so forth, Greek letters for resources, I, J, K, Roman letters for species. Okay? And so what MacArthur consumer resource model posits is one simple way of writing down the dynamics of these resources and species in one patch with just growth, no noise, and no migration. We're going to talk about growth and noise later on in the sort of classes, maybe in the 4th and 5th lecture. We'll talk about it. For the rest of the classes, we're only going to think about growth. Okay? So this is what MacArthur's consumer resource model says. Let me write down the equations and then I'll tell you how one thinks of them. Okay. So what have I written down? I've written down the dynamics for different species and different resources. For species, we are really saying that there are 2 things that are happening. Every species is able to extract different resources right? So it grows, it gets some sort of flux from each of the resources R alpha, but it only gets a flux proportional to this number we call Ci alpha. So you can imagine that C is some big matrix that kind of tells you for every species I, how much or whether it's able to eat resource alpha at all or not, right? So what we're imagining is, in some sense, the C matrix as, I mean, the way this is written, there are species on the columns, or on the rows, resources on the columns. Right? This goes from 1, 2, 3, alpha, all the way to M. We're going to call M as the number of resources. Similarly, there are 1, 2, 3, I, all the way to S species. And anytime you go to an entry, let's say 3, 2, if that entry is 0, that means species 3 cannot eat resource 2. If that is non-zero, species 3 can eat resource 2. And the larger that number is, the more energy it derives from eating that resource 2. And the Ci alpha is in some sense a matrix of what are called consumer preferences. Okay, and essentially what you're just doing in this sum is that you're summing over all the resources that you can eat and getting a total growth flux from that. This entire term is a term that tells you about, let's say, growth from consumption. Right? Remember, this is a sum over M resources. Yeah. So you get a bunch of growth flux or energy from eating all of these resources. If you're a living species, you're typically also subjected to some maintenance costs that you might have to pay just to be able to survive. And so that's this MI. This MI is some maintenance or some mortality or some death rate that you might face. In the face of that, this equation is saying something very simple. It's saying you consume a bunch of resources, you add up the total energy you get from all of them. If that energy is larger than MI, then what happens? You grow, and you grow exponentially at a rate that's given by this term in the parenthesis. If that's smaller than MI, then you die, right? And you die at a rate that's given by what's in the parentheses. And there's one more thing that can happen, right? They can both be equal, right? And at that point, what would happen? You would neither grow nor die, right? So at that point, like at a kind of steady state, if this species survives, you might expect it survives in a situation where all the consumption that it's doing exactly matches its maintenance or its mortality, right? And of course, in this kind of model, we also then have to think about the dynamics of resources, right? Because as species are consuming resources, they're depleting them, right? So this little thing over here is just about depletion, right? All this is kind of saying is that different species will then consume resources and deplete them. And maybe there's a slightly different matrix E as opposed to C that tells you about a species' ability to deplete a resource, right? It might be the case that you deplete some resource by some amount, get some energy from the resource which is slightly different, right? Like some resources you deplete a lot but get very, very little energy from. Some resources you deplete very little but get a lot of energy from. Some resources you deplete a lot and get a lot of energy from. So the fact that this C and E are generally different sort of captures that. And what's this little thing over here? So let's just think what would happen if we didn't have any term of this kind. What would happen to the resources? Eventually, they'd all just get depleted. And then if there are no resources anymore, then what happens to the species? They'll just die exponentially, right? So if you want to have a non-trivial steady state in which some species are surviving, you also need to supply the resources from somewhere, right? And this is sort of one simple way in which you can supply these resources. You know, there are many different ways in which resources might be supplied. What MacArthur was assuming is sort of the resources are themselves a little bit growing logistically, right? Did you guys do logistic growth or something of that kind when you were doing nonlinear dynamics in the tutorials? Yeah, is everyone familiar with that? getting a bit blank faces. Okay, so you did logistic map, right? But a continuous version of that is this. Different kinds of dynamics, but really it's very easy to see kind of and get intuition what should happen in these dynamics. So what happens when R is very, very small compared to K? Let's say you kind of start off and there are no species, so don't sort of worry about the consumers. What will happen to R? Will R grow or die? Grow or die? Grow, right? And will it keep growing? R will grow, grow, grow. At some point it will hit K and stop growing, right? So this is just logistic growth. You're growing exponentially. Then you reach some sort of number K alpha, you stop growing, right? This doesn't need to be the only way resources are supplied. Like I said, this is kind of one thing that we are assuming. In fact, this might not be the only way resources are consumed, right? This might not be the only way resources are depleted, right? So you might imagine a more general version of consumer resource models where this bit is replaced by some general function G of I. This bit, this supply bit, is replaced by some other general function, the H of alpha. This bit, this depletion bit, is replaced by yet another function, right? And you can kind of study what happens to a generic family of consumer resource models of that kind. Okay. Again, we're not going to do that in this class for simplicity, but you could imagine a whole family of models that don't have this assumption or that assumption and that are different, right? Okay. An NJ? Okay. Okay. Good. So EJ, so NJ or NI or NK are all our dynamical variables, which are the abundances of different species. So NJ would be the abundance of species J. Okay. EJ alpha, so R alpha will similarly be the abundances of resource alpha or the concentration of resource alpha. R beta would be the concentration of resource beta, and so on and so forth. And so EJ alpha is just an element of a matrix that tells you how species J depletes resource alpha, right? And what you're doing in this sum over here is, of course, the same resource might be consumed by many, many species, right? So you're kind of the total amount by which any resource alpha is depleted comes from summing over all the different species eating it, right? So this is N1 E1 alpha plus N2 E2 alpha plus N3 E3 alpha, and so on and so forth. Okay. Okay, yeah, please keep asking questions if something's not clear. This is important. Okay. And so, I mean, you can kind of see that in these dynamics, you have to worry about both what is happening to species as well as what is happening to resources, and the way, do species interact with each other in this model, by the way? Okay, let's take a little bit of a vote. Okay, so what we'll do is we'll use our hands. Thumbs up is going to mean yes. Thumbs down is going to mean no. Okay, and I'll count to 3. At the count of 3, everyone will put up their hands with a thumbs up or a thumbs down based on what you think. Yeah, we're not going to keep any kind of score. So please feel free to vote. It's kind of important that you do, because that's kind of the only way that you're thinking in the class, right? This will get us a bit warmed up. Okay. Later on, we're going to have questions that are going to have numbers in them, in which case we're going to use all our fingers. Okay. All right. So do you think species are interacting in this model? Ready? 3, 2, 1. I see some people with their hands up like this. I don't want you to answer right now. I just want you to think yes or no. Yeah. Okay. So I see lots of yeses, a few no's. I would say maybe, yeah, maybe like 60-40 yes and no. So maybe what I'm going to do is I'm going to get you to turn to your neighbor and the next maybe 10 or 20 seconds, try and convince them of why you think the answer is what you think it is. Yeah, and then we'll come back and have a discussion. Okay, let's maybe in the interest of time come back and take a little bit of a vote again. Okay, so we'll do the same thing again, flexing our thumbs, and take a vote again as to whether people think species are interacting in this model or not. Okay, ready? 3, 2, 1. All right, see lots of things turn, lots of no's turn to yeses. Some yeses turn to no's. That's interesting. Yeah, so maybe would someone care to say, maybe someone from the no side care to say why they think that there are no interactions between species? Anyone, it's totally fine. By looking at the equations, I depends on I only. Yeah, okay. So then there are no interactions between species. Okay, anyone care to counter that? R alpha, okay, so you're saying the dynamics of species I depends on R alpha. The dynamics of R alpha depends on the dynamics of every other species. Okay, and so Ni is indirectly dependent on Nj, right? So there's some weird coupling that's going on where species are not directly affecting each other, right? So there are no direct interactions between species, but they are eating away each other's resources, right? So what kind of interaction is this? Is it a competitive interaction, or maybe competitive should be like this? Anyway, is this a cooperative interaction or a competitive interaction? Ready? 3, 2, 1. It's a competitive interaction, right? Because species are kind of competing for the same set of resources. Okay? You might imagine a situation in which instead of depletion, there's another term here where species are also producing resources as they eat others, right? And that which other species can eat. And in that case, you might imagine there's to be some cooperation as well. Okay, good. Let's talk about negative matrix elements. So which matrix would you like to make the elements negative of? So C. Okay, great. So what would C being negative imply? Exactly. So some resources are toxic, right? And so I would say that's neither, I mean, okay, it depends on where the resources are coming from. In this model, the resources are only coming from outside, right? That just means that you don't think of them as resources, you think of them as toxins, right? But in that case, it's not about competition or cooperation, right? It's just about, oh, I just hate this particular resource, I just die, right? But indeed, if someone else was producing this toxin, then you could maybe talk about what kind of interaction that is. Good, maybe death of some species is food for others, which is also not assumed in this model, right? So again, this is kind of what I meant by, you could imagine a whole family of consumer resource models, which has slightly different assumptions about what is going on. This is in some sense, we will see the simplest kind of consumer resource model that we can write down, which has the capability of both being low dimensional and high dimensional and tractable and insightful. So we're going to stick with this kind of model. And actually, I'll just tell you, we're not even going to stick to this kind of model. We're going to stick to a simplification, a further simplification of this model, which we're going to discuss because we have only 5 classes. Okay, but everyone's clear that there are no direct interactions between species in this model, only indirect interactions mediated through resources that manifest as competitive interactions in this model. Other models might have other features. But what we're going to care about in these entire series of classes is not analyzing models of this kind where there are both species and resources. I'm going to stick to a simpler set of models where we actually only think about the dynamics of species. Okay? So in some sense, what we're going to do is try to get an effective model from this where we kind of integrate out the resources. Okay? We're going to try and forget the resources in some sense and focus only on the dynamics of species interacting with each other. So that's going to be an effective model. Okay? So to do that, what we're going to do is a common assumption, which is not always true, but we're going to assume it again for these classes to make things simple, which is that we're going to assume that the resource dynamics are very fast. In fact, they're much faster than the dynamics of species. And so what that means, this is a quasi-static assumption, everyone's probably familiar with this, but what would that mean mathematically in these equations? What should I do to make this assumption the case? Steady state of resources, right? So I always assume that R alpha dot is 0 for all resources. And we can ask what happens as soon as we do that. So if R alphas are not 0 at all, I can straightforwardly write down what my R alpha should be, right? My R alpha at steady state, which I'll call R alpha star, should be what? You can almost read it out from the equation, right? It's K alpha minus sum of Nj Ej alpha sum of J. Is that clear how we got here? If none of the R alphas are 0, I can take that out. Then I can just write down what R alpha should be, right? And you can see that it depends on Nj's, depends on K alpha, depends on Ej alpha. I'm also going to be a little bit lazy eventually and stop writing down what all these sums go over, with the hope that you will try and get in the mood that sums over J's or K's and I's will all be 1 to S, sum over alphas will all be 1 to M, where there are S species and M resources. Okay? So let's substitute this back into the dynamics of the species, and then we'll be done in one line of algebra. So if we substitute this, we get effective dynamics for Ni, which is Ni dot equals some Ni Ci alpha K alpha. I'm going to write the minus Mi over here for convenience. You'll see why in a second. Minus Ci alpha Ej alpha Nj. This sum goes over alpha and J. Is everyone okay with this? This is the simplest set of algebraic manipulations we'll do in the entire class. I just substituted R alpha into that equation and just rearranged terms around. The Mi came over here, right? Anyone confused about this? Okay, I can also do then one more thing, which is that I can sort of divide and multiply throughout by the following. I'm really just doing some manipulation, and you'll see sort of why. By C I beta, E I beta, sum over beta. And when I do that, I'm going to get the following. So outside, I'm going to get my Ci beta, Ei beta. Remember, the sum over beta now is going over resources, right? So it'll go over all the resources. Again, 1 to M. Ni. And over here, I'm going to get Ci alpha K alpha minus Mi. Okay, I didn't do anything special. I just multiplied and divided both sides by this. And why by this? This is essentially the coefficient here of Ni, right? Ni is, of course, special to the equation for the dynamics of Ni. I just want to kind of get the coefficient of that out and make it just 1. You'll see why in a second. I mean, actually, it's just a matter of convention. I end up with this, right? Now, all we're going to do, just to simplify this, this is something that, does it depend on beta at all? No, right? It's a sum over beta, so it just depends on I. This is some parameter R sub I. I'm going to call R sub I. What about this guy? Does this depend on any resources? Summed over all the resources, right? So again, this only depends on I. I'm going to call it some K sub I. You'll see why in a second. This thing over here, what does this depend on? This sum, remember, is over betas. This sum is over alpha, so it doesn't depend on resources. It depends on the I-J pair, right? So I'm going to call this sum capital A I-J. Not the sum. Maybe I should take the sum outside. Should be a little bit more careful. So maybe I'll keep the sum over J here, and here J is not equal to I. I'll keep the sum over alpha over here, right? So I summed over alpha. This is a term which doesn't depend on resources. It's some Aij, right? So this term, this whole equation, then I can write down much more simply as just some Ni dot equals Ri, Ni, some Ki minus Ni minus sum of Aij and J. And I'm just going to remember that this J goes over all species except the ith one. Why is that? I just had the ith term sort of sitting outside. Okay, so these R's, K's, and Aij's are some effective parameters that I got from essentially some underlying consumer resource model, right? But in the end, they're just some effective parameter, right? So I end up with a kind of a model where for every species, I need to care about this Ri, this Ki, and then this set of Aij's. And all of these have very nice interpretations. This R sub I is kind of like an intrinsic growth rate, or it has units of growth rate. Can we see that it has units of growth rate? Yeah, we haven't talked about units yet in these models. It'd be good time to talk about that. So what are the units of the left-hand side? It's some population density over time, right? Whatever the units of abundances are, which might change from population to population. Let's just think of them as, let's think of abundance as a fundamental quantity. Okay, so abundance over time. So this side must also be abundance over time, right? And so what should I think of as, this is my abundance. This is my Ri. All of these are what? They're also kind of abundance units, right? So what should the units of this Ri be? So this is abundance over time, abundance, 1 over abundance over time, or something like that, right? But fine, if you kind of think about, but it has some units of rate, but it's in sort of units of the abundances in some form, right? So it's some funny thing. But nevertheless, we're going to think about it as sort of growth rate of species I. This K sub I is going to be like a carrying capacity. This again is units of abundance, right? And you can kind of see why it should be a carrying capacity. If there were no other species, then what abundance would the species grow to and then stop? Ki, right? At Ki, you would stop again. So you can kind of see if there were no other species, then this becomes exactly a logistic model, right? If there are other species, it becomes a different model from a logistic model. But that logistic model, there it is, has a carrying capacity of Ki. Okay? And in these Aij's, what interpretation should I ascribe to them? These are some effective interactions, right? Remember, there are no direct interactions in this, there were no direct interactions in this model, but these are some effective interactions that we ended up with, right? And they are really telling us how species J affects species I. There's also some convention here that I kind of hid that I should expose now. I wrote Aij, right? So the way that I'm writing it and the way that I'm imagining it, is J affecting I? Yeah, remember I'm writing down the dynamics of N sub I, right? So it better tell me what's happening to species I, right? And you can see these are all the effects of species J, right? They were happening because species J depletes this resource, and that's how it enters into the equation for the dynamics of species I, right? It's the same sort of logic. This convention is going to be a little bit important, and it's a little bit non-standard, so please get used to it. This is what's common in the field. Aij is going to mean how I is affected by J. This model has a very famous name. It's a very famous model. And it's going to be the model that we spend the next 4 classes studying. Anyone know what it's called? Called the generalized Lotka-Volterra model. Maybe all of you have heard about the Lotka-Volterra predator-prey model. It's a model where there are 2 species, a predator and a prey. Predator eats prey, prey dies. This is a model where there's necessarily no predator and prey, right? This is a model where lots of species are competing with each other or interacting, right? And so in some sense, it's just generalized in many ways, right? It's generalized in the sense that it's not predator-prey model. It's also generalized in the sense that it's not a 2-species model. This could be any number of species that you want. This J could go from 1 to S. And in fact, as we will see, we'll want to send this S to infinity eventually, right? But this J could also be like, this thing could also go over 2 species, right? So this J, how many terms would be there in this sum if they were just 2 species? Ready? 3, okay. 1, 2, 3, 4, everyone knows how to point fingers. Okay, so the question is simple. If I were to study this model for 2 species, how many terms would there be in this sum? Ready? 3, 2, 1. 1, right? I mean, maybe as we go on in these classes, and I ask you a question when I do 3, 2, 1, try and use your fingers before you use your mouths, and then we'll get plenty of opportunity to use our mouths later. Yeah? Otherwise, of course, please feel free to speak. It's really to allow everyone to think. Yeah, so there's just one term in this sum, right? So this kind of model, like there are kind of 2 realizations here. One is that if you take a consumer resource model, especially this MCRM kind, there's a correspondence between these consumer resource models and these generalized Lotka-Volterra or GLV models. I'm again going to say GLV again and again in these classes. That's going to be generalized Lotka-Volterra. It's going to be hard for me to keep writing generalized Lotka-Volterra, so I'll just keep saying GLV. And this kind of correspondence especially appears under fast resource dynamics. This is a pretty big assumption. It's not always true. Nevertheless, what we're going to do is, for mathematical convenience, we're going to study the generalized Lotka-Volterra model for the entirety of this class. And you can treat as homework for yourselves basically every single calculation that we're going to do in these classes for the generalized Lotka-Volterra model, you can also do for the full-blown consumer resource model. Okay? And I'm going to leave that and going to give you a bunch of problem sets that are going to guide you through doing that. Okay? But every day where we're basically going to do a set of calculations, you can just go and ask yourselves, how do I do the same set of things, except where I don't assume that resource dynamics are fast? Then I'm going to have to deal with 2 sets of equations. For all the conceptual ideas that we're going to talk about, all the ways of talking about diversity, stability, and structure, they're all going to be possible for us to ask in this model. And so that's why we're going to use this model to kind of sort of study them. We're going to build in intuition for what happens with the generalized Lotka-Volterra model. Okay? Yeah, 2 questions. Yeah, go on. Okay, good. So the question is, can chaos be an emergent behavior of this model if we choose the right KIs and RIs and so on? Yes, and actually what we're going to see is that the AIJs are going to be the key things that matter. The AIJs are the interactions, and so indeed this model contains chaos in it. Yeah, there was another question. Okay, good. So we are not taking, so the question is whether the interactions between species, we are taking them to be reciprocal. Actually, we are not taking them to be reciprocal. Can someone say why? Does everyone understand what it means by reciprocal? The question is whether this Aij is a symmetric matrix or not. Right? Is Aij, is how J affects species I, equal to Aji? My claim is that's not true. What is Aij and Aji? Aij is this, right? Is this sum. Aji is going to involve Cj alpha, the sum over Cj alpha Ei beta, right? So different rows and columns. This is essentially this thing in matrix form. If you forget the denominator, which is just a number, is like Ce transpose, right? So if C and E are different matrices, then Ce transpose need not be a symmetric matrix, right? And so for that reason, this model is already non-reciprocal. Yeah? Okay, any other questions? Yeah. Any other special constraints on this matrix A? The answer is yes and no. We are going to assume that there are no constraints on this matrix A per se, but we are going to see that the model is only well behaved in a specific parameter region where some statistical properties of this A is satisfied. So we are not going to assume any constraints, but we're going to see that there's a well behaved region of this model and there's a badly behaved region of this model. Okay, so maybe in the 5 minutes that I have left, I'll just quickly, we won't go to do a full analysis of the two species version of this model today. We'll do that tomorrow. But let's just build some intuition for what the two species version will actually look like. Okay, so, I mean, we can sort of write it down, but the two species version is going to be N1 dot equals some R1, N1, A1 minus N1 minus A1, 2, and 2. Remember, there's only one term in the sum. N2 dot equals R2, N2, A2 minus N2 minus A2, 1, N1. Okay? And actually, to make it even simpler, because qualitatively everything is going to be contained in that model, I'm going to set both the R's to 1, and I'm also going to set both the K's to 1. Okay, this is just to make all the algebra simple. Nothing's going to change qualitatively. And again, you can try on your own later what happens and what changes when you have general R's and K's. Okay? I just want us to get a sense of what situations we're talking about when we're talking about these A's. So remember these two numbers, A12 and A21, they can both be negative, right? And so what kind of interaction are we modeling when both these numbers are negative? Is it cooperation or competition? Okay, we'll take a vote. Ready, 3, 2, 1. I still don't see half the class voting. Either everyone's confused or people are shy. Please don't be shy. I'm really not keeping score. It's not going to affect absolutely anything about the course, but it is definitely going to affect your ability to think through. Okay, then let me ask again. Both the A's are negative. Sign convention is whatever it is. Is it cooperative interaction or competitive interaction? Ready? 3, 2, 1. You can also do something in the middle if you think it's neither. Okay, yeah, it's a cooperative interaction, right? This is a mutualism or a cooperative interaction. Why is that? Exactly. So you can kind of see that every species has a growth rate, right? This term in the parentheses is like its per capita growth rate, right? Ni dot by Ni. And that growth rate is affected positively by the presence of these other species, right? That's a convention with which we've written these signs. What about when they're both positive? Is that a cooperative interaction or a competitive interaction? Ready, 3, 2, 1. The competitive interaction, right? Both species are competing with each other. And indeed, you can see that the way that we wrote down this consumer resource model, indeed, we are imagining all these entries are maybe positive, and so in the end, this is the situation that you might get or expect to get from if these A's were coming from this kind of model. Okay, there's finally one more situation, right? Which is what? One of the A's can be positive, the other one can be negative, and they can be either one of them. So I'm just not going to write both. And what kind of situation is that? Is that cooperative or competitive? Ready, 3, 2, 1. Or neither? Yeah, so if it's neither, what kind of interaction is it? Yeah, it's kind of a parasitic interaction, right? One species is benefiting from the presence of the other one is getting inhibited by the presence of the other. It's kind of like a parasitism. Okay. What we are going to spend our classes talking about is this kind of scenario. Right? There are many reasons for this. One of them was we were imagining things coming from an underlying consumer resource model where there's competition. Another is that is nice. I like that. The other is that also, of course, there's a big tradition of studying competition. Okay. One can, of course, study cooperative models. And again, I won't have time to do that in all of these classes, but I encourage you to take a look at sort of all of that later. Yeah. In this one, in the competitive one, is any of them a prey and any of them a predator? No, right? This is not a predator-prey model per se. Which one is closer to a predator-prey situation out of the three? It's the third one, right? So this one is more predator-prey. This one is not predator-prey. This is just two species competing. Okay, all right. So we'll stop here. Next class, we'll pick off where we left off. We'll start off with the GLV model. We'll study it for two species. We'll get a feeling for what the dynamics look like, what kinds of qualitative outcomes can happen, whether they match experiments or not. And then we'll go on to asking how do we extend what happens not for two species, but for many species. Okay, in the classes that go on, we're going to forget for a second about the fact that all of this comes from an underlying consumer resource model. We are just going to assume that this is the model that we started off to begin with. Okay? All right. So I'll see you all tomorrow.